Welcome to the Peak Performance Podcast, your guide to optimizing your life one health tip at a time. This podcast is designed to entertain and inform, not to provide medical or health advice. Always consult your doctor when it comes to your personal health or before you make any changes to your diet, exercise, or lifestyle.
Ladies and gentlemen, welcome to the Peak Performance Podcast. I'm Coach Kyle here with Dr. K and Dr. Keith Ellis, PhD. We're very honored to have him on our show today, and we are going to talk about long COVID. This is kind of risky, being online talking about COVID because, we could get shut down, shadow banned.
Well, so I think we can talk about COVID. We're allowed to talk about it. It's just like we can't promise that we're going to cure COVID using alternative methods, which we're not going to. We would never promise that.
Yeah. So this is more about Dr. Keith Ellis's approach to alleviating the symptoms of long COVID, which I think, I had a discovery call with him previous to this, and I think it's a very innovative approach. So before we dive into kind of the specifics of the approach, I'd love to ask Dr. Keith just to kind of go over his background, who he is, what he does, and how he arrived at this long COVID thing.
Hey guys, thanks for having me on. So my background is one that there are a lot of people out there with it, but I don't think there's a lot of people that are working in the kind of natural health and wellness space with my background. I'm a medicinal chemist by training. I actually started out as an organic chemist, and then I trained in medicinal chemistry, and I'm an associate professor of medicinal chemistry. And kind of what I've done for my career is I took my training in organic chemistry and how to make molecules, and I actually started out as a natural product total synthesis chemist. So I've been kind of intimately involved with natural products really since I was in grad school. And I paired that with medicinal chemistry, and medicinal chemistry is the academic discipline that basically does what we think of as drug discovery in pharmaceutical industry. So medicinal chemistry departments, we basically train our students to go out and do drug discovery in pharmaceutical industry as that kind of first phase of trying to develop new drugs to treat human diseases. So kind of historically in my lab for most of my academic career, I've worked in what I think of as early lead discovery. So looking at disease states, looking at kind of the latest literature that's out about what are the mechanisms at the kind of molecular level behind a disease state and what are new, what we call molecular targets, right? Proteins and other molecules in the body that we can use to actually modulate and try to help alleviate symptoms and root causes of diseases. And then actually start the process of developing small molecules that can be eventually developed into drugs for those conditions. So that's what I've done for most of my career. I started out working a lot on antibacterials and then shifted over to really working more in the oncology area for most of my career. But that's kind of the skill sets I'm bringing to the table are kind of that molecular interface between biology and chemistry in the body of where we start really examining disease pathophysiology, right? The actual underlying molecular mechanism that's causing the disease, and then how we turn that information and use it to actually start developing new drugs.
So you're not the guy that is like testing the new drugs on rats. You don't go that far.
No, that's way farther down the line. I don't do animal studies. I my main skill set is really making molecules. It's synthetic chemistry. So we're trying to identify those early stage small molecules. We do some assays in my lab, particularly like biochemistry ones in a test tube, right? We've done cell assay work in my lab, and that's about as far as I go. And then I get collaborators typically that are trained more in animal studies and preclinical work. And I collaborate a lot with physicians as well, and physician scientists that do those later stages of drug development.
Yeah, I think it's interesting that you do that kind of front end work, because when people think of like a drug process, I don't think a lot of times they think about, hey, let's look at the literature first and let's look at what could work and mechanisms before it gets to the first stage in the FDA approval. A lot of people just think, hey, we have something, let's test it on a rat.
Right, and that's this kind of early work that has to be done really for any drug. Is you have this kind of of period where there's a lot of basic science research going into the molecular mechanisms of the disease state, tying symptoms to mechanism. Eventually, we collectively, science accumulates enough of a body of work that you can say, okay, here's actually some potential molecular targets, some potential proteins or other molecules in the body that we can actually develop a drug for. Because the other thing that people don't I think appreciate all the time is developing a drug takes a long time. It's typically a 10 to 15 year process, and we're not kidding when we say it's expensive. Everybody talks about billion-dollar drugs and kind of vilifies big pharma with that, but at the end of the day, going through the drug discovery and development process is actually super expensive, and it's a high-risk business endeavor because at any point, including up to like stage 3 clinical trials, your molecule can fail and never get approved, and you just basically lost all the money you invested. So I like to do this early work because there's been more emphasis over my career of doing better work early on to try to minimize those failures later by doing really good work up front.
Yeah. I imagine it's kind of like in the discovery process. It's, I've done a lot of work in real estate in my career, and when I'm looking at like a land deal or development deal, I might look at 10 and one might be good. So I imagine it's a lot like that. You look at a bunch of molecules, and then by the time you do all the research review and mechanism review, you're like, oh, well, most of these don't work.
And that's pretty much it. I mean, we call it lead discovery for a reason, and it's kind of like leads in other business endeavors like real estate. You have to, once you pick a molecular target, we tend to validate it. And that's usually where I step in, is once a molecular target's been validated, right? That means like there's really good foundational science showing that particular molecular target is really important for the disease state. And if you modulate it in a certain way, you actually affect the disease state and the symptoms that come from it. So once you have that, then you do what you're called lead discovery, which would be kind of like finding leads in real estate, right? You have to start with hundreds, thousands. When pharmaceutical industry does this, they start with typically a couple million compounds and do a high throughput screen to start finding the initial hits, because the attrition rate increases as you go through that process. So you do have to start with a large number of compounds. That you're going to lose 90+% of them along the way.
Yeah. So there you go. I look at 10 land deals, but he looks at millions of molecules. So how did you make this jump from, orgo, medicinal chemistry to, hey, long COVID is something I want to focus on? Where did that come from?
This is one of those things that kind of, I started this as a kind of an academic project the way I would any other, wanting to look at long COVID. And then it just took this radical turn, and I went where science took me, and it just took me to a very different place this time, right? So early 2023, COVID had been around for a while. Long COVID was starting to be a real problem, and there didn't seem to be a lot available for it, right? With acute COVID infections, we understand how to clinically manage acute viral infections. There's antivirals that are being used for acute COVID infections. We seem to have gotten past that phase of acute COVID where a lot of people were getting hospitalized and dying. Not that it's not still a significant clinical problem. It seemed to got better as the time went by because the virus mutated and wasn't quite so nasty and caused so many problems. But by early 2023, long COVID was really emerging as this problem. And there didn't seem to be a whole lot people could do about it. And that kind of really interested me. And I figured it'd been 3 years since the beginning of the pandemic. And I knew kind of from following the background in science that a lot of grant money was going into COVID and long COVID, a lot of publications. The publication numbers in terms of the volume of publications has been off the charts in the COVID era since 2020, right? So I figured, okay, let's start doing what I normally do to start a project, to start looking for the mechanisms that are underlying the disease state, to start looking at, okay, what are the molecular targets out there? Have they been validated? Because Oftentimes I'll start a project when I think something looks good, even if it's not fully validated, because I want to get ahead of the curve a little bit. And having those tool compounds sometimes is just as important in the early phases as having a real lead that has a chance to make it all the way through clinical trials. So I started the project the way I normally would an academic drug discovery project. And I kind of did the review of the literature and looked at everything that was available. And at the end of that, I was surprised, I was shocked, and I was a little kind of both frustrated and a little bit like angry when you look at something and you're like, wow, this really shouldn't be this way. Because taking a step back and looking at the literature that was available in long COVID, there weren't really good starting points for where I typically enter the process and start early lead discovery in a disease state. There was a lot of volume of information being published. A lot of faculty are getting grants to do the work. They're publishing very good work, and I don't mean to disparage anybody's publication. All the science that's being done is super high quality. It's super sophisticated. What I was frustrated by was there was somewhat of a lack of focus in the literature, and nobody was really trying to put the puzzle pieces together to really flesh out what's the pathophysiology, what are the mechanisms, and what are some things that we can use to start actually developing things to help people. What I do is I want to actually start that process of developing something to help people, and the literature was not really focused on that. So I had to take a step back, and that's kind of when I pivoted and changed directions kind of in reaction to that.
So my first question to that, and this may feed more into it, is how are we defining long COVID?
Keep in mind, I am a scientist. I'm not a clinician. I've spent a lot of time in the literature. The definition of it is not as crystal clear as I think everybody would like it to be. There are more kind of diagnostic criteria. There's almost criteria for exclusion. There's no checklist of if you have A, B, and C going on, you have long COVID, right? So part of the basic definition of long COVID is it is symptoms and effects post-acute COVID infection that linger on beyond 3 to 6 months. So the timeline typically starts with an acute COVID infection. People will get maybe most of the way better, but they still have symptoms that linger on 3 to 6 months later. And then typically in that window, they actually start developing new symptoms that are not what we typically associate with an acute COVID infection. So at that point, then, once you are in that time window post-acute COVID infection, then it becomes looking at kind of the constellation of symptoms that someone has, and it's diagnosing it that way. So there has to be kind of that proximal cause of you got COVID, it's on this timeline, and then you have all these other symptoms that fit the profile, but the symptom constellation or array for long COVID is somewhat variable and somewhat large.
Right. And so, well, I think that leads into why there's no like concrete science data on it, right? Because no one can agree on it. And then the secondary thing I've noticed, so a lot of my patients that come in with long COVID, I've started testing people for Epstein-Barr, reactivated Epstein-Barr, and that comes up as positive. And so when we focus on solely treating the Epstein-Barr and ignore the long COVID and the Epstein-Barr goes down, their symptoms go away. So I think part of it is that long COVID is being misdiagnosed as other things. And now just like anytime you don't know what's wrong with someone, they got sick after having COVID, you're like, oh, you have long COVID.
That's what I was going to bring up as well. Like if we can't agree on a specific definition, how do we know it even exists? So like being quite frank with you, I'm a researcher as well. I'm a doctoral trained researcher. So if there's no definition, how do we know we're not tracking something else entirely? Like for example, it could be long COVID is just the people who have had COVID and they got reactivated Epstein-Barr, or one of 400 other things.
The difficulty here is what everybody wants and we don't have is there's no like clinical lab tests that you can run to actually say, okay, you have long COVID definitively, right? And I think the frustrating part for both physicians and for patients is you tend to run labs and a lot of them look mostly normal, right? So it's hard to even understand whether something's going on with these patients. They're sick, but when you run standard clinical labs, a lot of times they will look very normal. And what appears to be going on, you brought up Epstein-Barr, is long COVID has joined this kind of pantheon now of what is collectively being grouped together as kind of post-viral reactivation syndromes, right? So it's Epstein-Barr, chronic fatigue syndrome is thrown in there. A lot of times you see West Nile and Lyme, as well as long COVID, kind of all grouped together. And it's reasonable to do that because at the kind of top line symptom level, they all have very similar symptoms, right? So it doesn't surprise me that you're seeing people come in, they have reactivated Epstein-Barr, and when you treat that, it does alleviate their symptoms because the symptoms are all so similar. It's difficult to conflate. It's easy to conflate the various viruses that are reactivating. So you can start treating the symptoms. Now, for some of these other ones like Epstein-Barr, we know how to treat the symptoms. For what really is long COVID, if it really isn't Epstein-Barr and is actually post-viral reactivation syndrome that is caused by COVID, so it would be long COVID, we just don't have enough research and information yet on what to do to actually help those people's symptoms. There's starting to be some of that in the literature, and that is what I kind of focused on and moved forward with.
I think the second problem is people have different symptoms. One person with, and I'm just going to say long COVID, has headaches and nausea. The next person has tinnitus. The next person lost their sense of smell. The next person can't keep anything down, has chronic diarrhea. The next person has severe debilitating fatigue. And so, yeah, we're just grouping all these things under long COVID. But then the first problem becomes, how do we make a quote unquote medication to treat these symptoms when they're all different symptoms? And if we start getting to symptom management, then we're not really even treating anything. We're just managing symptoms, which is what, with the Longevity Center, most people in this field try and get away from is symptom management.
And I'll play devil's advocate on that. Coming from a biohacker perspective, which is kind of my stance, I'm more of a throw a bunch of things at the wall and see what sticks. If we can find something that improves the symptoms across the range of symptoms for the majority of people, I don't care what it is. I don't care if it's Epstein-Barr reactivated. I don't care if it's Nile. I don't care if it's long COVID. If it works, and it's a solution for people. And I think I can kind of see that to some degree in your worldview, Dr. Ellis, because it's you seem very pragmatic in your worldview. And I think that's another problem. A lot of researchers are not pragmatic. They just want to learn and publish and not necessarily have a direction come from it.
Okay, what you said is exactly right. The other frustrating thing with long COVID is when I've been talking to the patient population, again, I'm talking to them a little bit differently than you are. You're treating in clinical practice. What I've seen is there's kind of core symptoms that most people share. Mm-hmm. And then there's this like larger constellation of symptoms, but that some people get, some people don't. Mm-hmm. But at the end of the day, all of the symptoms cross physiological systems in the body, which is why medicine has such an issue dealing with it, because we've siloed medicine into various physiological symptoms in the body. That does actually lead towards more wanting to treat symptoms, but then treating them on a system by system basis. And you're right, Kyle. I'm more of that guy. When I'm doing these things, when I'm looking at them, number one, I'm looking at molecular mechanisms. And my thought process is, I don't think it's totally unique, but it's different than I would say the majority of science, because I'm thinking like a chemist. I'm thinking at the chemical level, even in a biological system. So that's the kind of level of detail I'm looking at. So when you're looking at that molecular level, you can do one of two things. You can say, okay, what's the mechanism that's causing the symptom? And how do I alleviate the symptom? Or you can say, well, what's the mechanism that's the actual root cause for the disease state, where the system is downstream, and that symptom might be caused by propagation through all kinds of signal transduction cascades. And it could be 20 steps from real causative mechanism physiologically to symptom. And so when you look at it mechanistically, I drift towards wanting to look at causative mechanisms, which is why I focus really on pathophysiology. And that's kind of where I jumped into long COVID. And what I am doing is different, because a lot of people that are doing long COVID research right now in COVID, they're basically, if they worked on neuroinflammation before COVID hit, they've now pivoted, and maybe they were working on neuroinflammation in some kind of autoimmune disease. Now they've pivoted and they're looking at it in long COVID or another post-viral reactivation disease. But they're basically taking their same tools, their same skill sets, the same maybe mechanistic pathways they were examining in another context and just applying it to COVID or long COVID. When I saw the state of the literature, I kind of took a step back because being on the front end of drug discovery, I'm not married to a specific mechanism or type of biology or molecular target. I'm kind of equal opportunity, right? I'll take anything that looks tractable and then move forward to try and make something that will help people. So that's kind of how I have a little bit of a different perspective to begin with. And then once I saw the state of the long COVID literature, that really forced a change in thinking for me, because basically the literature is not ready yet to do traditional drug discovery to come up with new pharmaceuticals. There's just not enough science and data yet.
So I'm on the edge of my seat now. You've done all this research and you found the problem, right? This is how good research starts. We find the problem and then we work to find, the mechanisms, and then we move towards a hypothesis and then testing. So what'd you come up with?
Went through the literature first time and I really couldn't find molecular targets the way I really wanted to. I kind of took a step back, and at that point I had gotten to know a couple people with long COVID. One of my good friends had long COVID and had a really bad experience. Got shuttled around from doctor to doctor, got no help, was really having a bad day-to-day impact in their life, right? So I kind of took a step back and I thought, well, I have all this expertise. I'm a scientist. What can we do right now for people, starting with where science is right now with long COVID? And I kind of went back to literature. I just asked a different question with a different mindset of not what can we do that will give us something in 10 to 15 years as a drug, but what can we do now just to help people, whether it's a drug or not, right? So in asking that question and approaching literature that way, that's when I found the great work that's been done by a bunch of scientists. It's called the Microclots hypothesis, right? And it's this hypothesis that microclots form as the result of COVID spike protein binding to a protein in the blood called fibrinogen. Fibrinogen is the natural protein that forms clots as part of the coagulation cascade, right? So the microclot hypothesis says COVID spike protein binds to fibrinogen and causes a change in conformation to an unnatural conformation that then leads to polymerization and formation of microclots. And microclots are sticky, they're unnatural, they stick to red blood cells, they stick to and activate platelets, so you get platelet hyperactivation. They can cause an autoimmune response. Most people are familiar with Alzheimer's, right? Everybody knows that Alzheimer's is caused by people get plaques in the brain that we typically call amyloids of the A beta or tau protein, right? But they get these unnatural plaques of protein in the brain. That's what causes Alzheimer's. The microclots hypothesis basically says viral protein binding to proteins in blood causes amyloids of fibrinogen, amyloids of blood clotting proteins in the blood. So you end up with these unnatural plaques that are these unnatural clots in the bloodstream that the body has a really hard time clearing. And those microclots that form as a result of binding viral proteins are responsible for all of the downstream molecular level and physiological effects, all the way down to the symptoms.
Let me give a shot at the fifth grade explanation. Let me see if I'm understanding you right. So when you get COVID, you get these things that end up staying in your blood for a long time. They hit other things in your blood, which causes them to mutate into tiny little monsters that screw up your systems all downstream.
Exactly. Yes.
Okay. That's the simple version. Yeah.
That's why we keep him around. He's our translator to people.
That's good. I need that.
No, we've found that listeners, some of our listeners really enjoy the heady scientific approach, but a lot of our listeners enjoy basic explanations.
Here's your really good fifth grade explanation. Basically, viral monster interacts with and bites your own monster in your blood, basically turns it into a zombie, and it goes after you.
There we go.
That's really what it comes down to.
Okay. And so that's all immune system based, right?
So, well, this is the interesting part. So the microclots are really circulatory system based, but they have broad effects across multiple systems, right? So When these microclots form, they in the circulatory system, they cause inflammation in the blood vessels. They do cause an immune response component, right? The theory is that it's one of the things that accounts for the autoantibody formation, where you make antibodies that are unnatural and attack yourself. It's in response to microclots, right? Because they're in the circulatory system, the microclots can affect cardiac function, so that would and respiratory function. So that's where the shortness of breath and the tachycardia.
That's why blood comes out looking like honey. It's really thick.
Right. It comes with a thick. It's thicker, but there's no change in tests like the clotting time, because you're not affecting natural clotting. You're just making these unnatural clots. It affects tissue oxygenation, so that contributes to both the fatigue, kind of generally, where the microclots impair oxygen delivery to tissues. It starts accounting for the CNS effects. The brain fog is, again, poor oxygen delivery to the brain that causes the brain fog and the molecular and the mental clarity issues. And it can also go as far as accounting for things like depression and anxiety, because the microclots, by affecting platelets, affect serotonin levels. And it can account for things like leaky gut and gut dysbiosis, because that's thought to be caused by viral persistence.
So having said all that, here's my question. I don't know how you're going to feel about this question, but why do we need a new drug when we have methylene blue? Are you familiar with that one?
I've heard of it used. What's the actual mechanism for methylene blue?
Methylene blue has multiple different mechanisms, and I'm going to talk just very simply so we don't lose our audience. But one of the things it's really good at is binding alpha, beta, and tau proteins in the brain. So we use it for Alzheimer's, to help treat Alzheimer's. The second thing it does is it's really good at helping cell. It's used for carbon monoxide poisoning, right? And what it does is it makes your cells more able to accept oxygen. And that's just a very simple. I'm staying away from the heavy science on it. But it seems like that would be treating the two main things that you just mentioned, which are the decreased oxygenation to cells and the buildup of these amyloid plaques.
Well, and I'm going to tag on there before I let you go and add on EBOO, another thing, a shameless plug that we offer. So EBOO is basically you run your blood through a dialysis filter, which in theory should clear the microclots, and then you get ozonated, which results in a massive increase in oxygenation throughout the body.
Yep. And so, that's what we used to treat long COVID is EBOO and methylene blue. But methylene blue, it's like $50-$60, and anyone can buy this over the counter and take it. I think it solves the two main issues, and I've been using it to solve the two main issues for a while now.
Yeah. Now, EBOO is not necessarily a wide-ranging solution for people because it's not exactly a cheap treatment. We have the best price in the country, but it's still, $800, $900.
Okay, so let me break this down, because I see kind of three pieces here. So let's start with methylene blue. So I had not heard you could use it to treat amyloid plaques of A beta and tau. That's interesting. That's kind of cool approach. I like that. I have not seen any literature of people. So amyloids that form in the bloodstream in long COVID are not made of tau or A beta, right? They're made of the protein fibrinogen, and other blood-based proteins, right? Mm-hmm. So the question I would have kind of mechanistically is, does methylene blue have that effect with A beta and tau amyloids because it's specific to A beta and tau, or because it is a mechanism for helping disrupt amyloids in general? So that was the question I would ask to really definitively nail down, okay, is that really a potential mechanism to address microclots, which are amyloids of fibrinogen? It's interesting that methylene blue helps with tissue oxygenation. I think that's great. I was not aware of that. In the work I've been doing, I used a similar approach. Like I used beetroot extract for nitric oxide to boost oxygen delivery. That's a component I put in my product for kind of the same reason, right? Trying to boost oxygen delivery to the tissues to help with the fatigue, with the brain fog, and with general recovery. So that makes sense. I didn't know methylene blue had that effect as well.
It sounds like that's basis of what the kind of research you do, right? Is you investigate whether this thing that does this thing already can now work for this new prospect.
So, and the interesting thing, so I'm used to thinking about methylene blue as a dye, right? As a chemist, that's the way we're trained, right? That's one of the dyes we are taught and deal with. What's interesting is, so we talked about testing for long COVID, and there's no clinical test, but the Microclots researchers that are working in this area, they're really good. They developed a research lab assay for Microclots for long COVID, right? So they basically took spike protein they made in the lab. They took blood samples from people that they got before COVID broke out. So they were certain that there was no COVID viral contamination. And when they add spike protein to the blood samples, they can form microclots, and they visualize the microclots using another stain, right? A fluorescent stain called thioflavin T, which is used typically to visualize in the research lab to visualize amyloids. It's known to bind to and be able to visualize under a fluorescent microscope amyloids. And that is also used for A beta and tau amyloids in Alzheimer's research. So part of me is wondering how similar the structures of thioflavin T and methylene blue are, and what those real effects are. Now, in long COVID, when you do the thioflavin T assay, that dye doesn't break up the microclots. You can visualize them, but they stay present, right? So, but it would be interesting to kind of do that experiment to see if the Microclot research team, they also took patient samples from long COVID patients and ran their assay with thioflavin T on it, and they can visualize the microclots. It'd be interesting to do the same thing with like methylene blue.
Yep.
Add that in, see if you can visualize the amyloids that way, whether that's a good stain for amyloids, and B, whether you can actually disrupt the amyloid complexes that are the microclots in long COVID. Those would actually be really interesting experiments to do. That would be that are like
just put our names on the back end of the paper. Just put our names on the end of the author list, and send us the cred.
And some of these, and this is what's frustrating, is some of these things to do, like, they're logical, they make sense. We came up with this in like 5 minutes of just talking about it, but the data is not being. But these experiments aren't being done and published in the literature, and it makes you wonder. It's like, why aren't some of these more common sense, logical things being looked at and being done, early?
Well, I think it boils back to the very beginning of the talk, and all the things you're talking about is like you're looking through all these scientific papers, looking for like a concrete explanation and science and this and this and this and this, when no one wants to take that step back and admit that, yeah, we don't have a scientific answer yet. Let's think about it like we're in third grade.
Yeah. No, everybody is so far in the weeds of their own sophisticated science that nobody's taking a step back to really look at those things.
I say this about medicine, and it probably applies to research as well. Medicine has become so specialized that if you break this finger, it's one doctor. But if you break this finger, it's a different doctor. And I think the same thing's applying to a lot of fields because people need jobs. And so you just keep diving in further and further.
Well, it is, and to a certain extent, and I think this is true in both medicine and in kind of science, especially academic science. The incentives to do things have changed, right? You're less, if you're a long COVID researcher, your immediate worries are, your students you have working in the lab, getting data to publish whatever the next paper is, getting data to get the next grant, right? You're often not taking a step back to say, okay, what's the thing that will help things the most? I did want to make a comment real quick on the EBOO and mention that there has been work done in long COVID on using a process called apheresis, which I think gives a similar result to EBOO. And it has been shown, what I saw when I looked at apheresis is people had trouble accessing it. There were not a lot of clinics that did it. It does have a little bit more of a cost associated with it. But I'm curious, are you guys seeing good results in terms of how long the effects last and people feeling better? Because one of the issues with apheresis was if you don't deal with viral reservoirs that are on board that are kind of causing the microclots, then it's a temporary effect and you get sick again after a period of time.
You hit my favorite question. The key difference between plasma apheresis and EBOO, the plasma apheresis is filtering and it's changing out your globulin, right? Whereas EBOO, it's filtering using a dialysis filter, so it's filtering out what's readily available in the blood, but we're infusing ozone the whole time. And what the ozone has the magical properties of, it's killing them.
So that's our approach, but you've heard enough about us. You didn't come here to talk about us. Although we got to get our team with plugs in.
Yeah, no, I think what you guys do is really good, really interesting. And at this point, everybody that's helping anybody with long COVID kind of has to stick together because there's not very many.
Yeah, no, agreed. So what did you come up with? After looking at this microclot hypothesis and kind of seeing what could help solve that, what'd you come up with?
Sure. So in the literature, the microclot scientists that were working on the hypothesis, they actually took it to the logical conclusion. They said, okay, these are microclots. And it's been known that if you use a single anticoagulant, it doesn't actually really help people in long COVID. But what they did was they took a pharmaceutical cocktail known as triple anticoagulant therapy. It's got 3 anticoagulants in it. They ran a small study. It was about 90 patients with long COVID. And depending on how long the people were on it, because they grouped them into a mild long COVID and a severe long COVID, but by the end of 4 to 6 months, about 80% of the patients with long COVID recovered. And the really interesting thing about this study is they ran microclot testing on the patients before and after. So before, everyone had microclots. At the end, the ones that recovered, their microclots were gone. So it basically verified the hypothesis that triple anticoagulant therapy is a good therapy for long COVID. The issue is after that came out, patients still couldn't get triple anticoagulant therapy because there's a couple issues with it. One, it's 3 anticoagulants together, so there's a significant safety risk with the side effect of bleeding. Again, the doctors are kind of hesitant to prescribe it, both because of the safety risk and also because there hasn't been a large randomized, placebo-controlled clinical trial. There's no big evidence-based study, which physicians are really wanting now to prescribe it. And because it was 3 pharmaceutical drugs for long COVID that aren't approved for that, there was a cost issue. So the insurance companies wouldn't necessarily cover it.
What were the 3 medications?
It's apixaban, is an anticoagulant.
Eliquis?
Yep. Low-dose aspirin.
Okay.
As a COX-1-2 inhibitor.
Yep.
And then clopidogrel, which is a P2Y1, P2Y12 receptor antagonist.
Okay.
So seeing what was going on, I basically said people needed a safer, more accessible, without a prescription, of inexpensive version of triple anticoagulant therapy. So I took my expertise, my training in mechanisms of action and pharmacology and my natural products background, and I basically reverse engineered the pharmaceutical cocktail into a natural version. So I substituted each pharmaceutical drug with a natural ingredient that has the same mechanism of action, because at the end of the day, it's the three mechanisms of action together that help break up the microclots. So that was really what I did, was I made basically a natural version of triple anticoagulant therapy, and that kind of led me. I had to get it to people, so I formed a company and had it manufactured, and have been selling it to my long COVID audience, and people seem to be getting good results with it.
So, 2 questions: what's in it? And number 2, do you have any evidence? Have you conducted a small scale trial on the effectiveness of it?
So what's in it? It's got turmeric in it, organic turmeric, which is curcumin. That's a factor 10A inhibitor. It's the same mechanism of action as apixaban. It has willow bark extract, which is also a COX-1-2 dual inhibitor, which is the same as aspirin. And it's got an extract from traditional Chinese medicine called danshen. That danshen has 2 natural products in it called, the names are escaping me right now, but there's 2 natural products in there that act as P2Y1, P2Y12 receptor antagonists. So it's a 1-to-1 replacement of a natural ingredient for the pharmaceutical mechanisms of action.
Is there a bleeding risk with it?
It's fairly low, right? Turmeric does have some bleeding risk. Like if you're going to go in for surgery, you're often told to discontinue taking turmeric a couple days beforehand. but the bleeding risks are fairly low. Just with those ingredients in general, especially at the amounts that are in my custom formulated supplement, there's not really bleeding risk associated with those doses. So it's actually a pretty safe product.
Would that make it in turn less effective?
So it's one of those where you are trading potency for a safer profile. So is it going to be less potent than the pharmaceuticals? Yes, because they're almost too powerful in this case, right? So yes, it's going to be less potent than the pharmaceutical cocktail. It's still, the mechanisms of action are going to get the job done. You are going to have to take it for longer, right? But you're making the trade-off of safety and accessibility for having to take it longer and being able to take something that's natural and doesn't have some of the risks of the pharmaceutical drugs.
How does taking it longer work? And so correct me if I'm wrong, this is my thinking about it. When you take the 3 medications, they instantly come in, they kill off all the clots that could form, and then you eventually stop taking it and washes out. So how is it that taking a lower dose of those things, they wouldn't be able to cover, in theory, all of the clotting that's happening?
That's actually kind of not the way either the natural or the pharmaceutical version work, right? So when you think about using an anticoagulant combination in this context, what you're doing is you are slowing down natural clotting. You're slowing down the formation of microclots by taking either version, okay? And in doing that, then you're giving the body more time to naturally clear the microclots. So the issue with microclots is because they're in unnatural conformations, they need more time to break down. The normal processes for clot dissolving in the system are much slower on these unnatural clots. So what you're doing is you're throttling back the coagulation cascade, basically, and you're impairing microclot formation, new microclot formation, to let the body clear the microclots over time.
So you're solving the bottleneck.
Pretty much, yes. But if you're using a less potent natural combination, you're assuming that you're not slowing down new microclot formation as much, so it can take a longer time to clear the microclots that are there.
So basically, to summarize, at 5th grade, if your body is making microclots at 50 an hour, it's clearing them in long COVID patients at something less than that, 40 an hour.
Yes.
And so the pharmaceutical would drop that 50 production down to like 25, whereas the supplement would drop that 50 production down to like maybe 35.
Exactly. Yes.
So it allows over a course of time for the body to catch up, break them down, and then eventually it will get its reservoir to 0.
Exactly. And you know, I think over time, I would expect, whether you're on the pharmaceutical or whether you're on my natural version, over time you should be able to stop taking it once all the microclots clear, presuming that you clear all your viral reservoirs and any source of the spike protein.
Well, and that's what was going to be my next question. How do you clear the viral reservoir and the spike protein?
People are still working on that. I actually have another thing I'm working on, because there's some disagreement still as to where the viral reservoirs are occurring. One of the hypotheses that I think has really good data behind it is it's occurring in the GI tract. It leads to a lot of the gut dysbiosis issues and the leaky gut. And basically, it's persisting in the microbiome in the gut. So you have to solve that issue in parallel with clearing the microclots. And that's something I'm actively working on kind of for the future.
Got it. Cool. So there's more to come. Stay tuned.
Yes. My long-term goal is really to take this approach I'm using and use it, to solve multiple problems in long COVID, but then also to address some of these underserved, underrepresented chronic diseases that need more help and aren't getting it from kind of mainstream scientific establishment and big pharma. So looking at things like chronic fatigue syndrome and West Nile and Lyme to see what we can do with the data that's available, because nobody's really working on actual pharmaceutical drug discovery for a lot of these disease states.
So one more idea for you. And I know we need to wrap up soon, but methylene blue has been shown to deactivate and or kill viruses. So running methylene blue orally along with something like the triple anticoagulant therapy,
Absolutely,
could help reduce that viral load as well as the microclots.
yes. The viral load issue, I think it's kind of two things. It's the viral persistence, but then it's clear in a lot of people that are sick with, whether it's long COVID or Epstein-Barr or anything else, their immune systems aren't functioning properly. They're not fully able to kind of clear the virus and whatever is going on. So anything you can do to start having that reduction of viral load and an antiviral effect in vivo, whether it's boosting or trying to kind of rebalance the immune system to do it naturally, or other compounds that are coming in to help have that antiviral effect. Anything you do like that is really going to help people get better, because that removes the source of the problem, and then you have to deal with any kind of persistent mechanisms that are going on, like microclots, that wouldn't necessarily disappear just because you clear the virus.
Gotcha. Okay. Well, Dr. Keith, thank you so much for coming on and talking to us about long COVID and your approach to it. Where can people find you and find this triple anticoagulant supplement?
Sure. So if they're interested in the natural version of triple anticoagulant therapy, I called the product CircuGuard. They can go to CircuGuard.com. They can order it there. There's a link there to the kind of the full product page that lays out exactly what's in it and why it works and all the mechanisms. In terms of finding other things I've done, if they go to KeithCEllisPhD.com, that is like the page of links to find everything. So when I did all this research, I really wanted to publish it in a way that was accessible to everybody. So I published all of this on a Substack, looking at, examining, asking questions about microclot hypothesis, kind of validating it, and then all of the design aspect of my product. I published all that on Substack. So if you go to KeithCEllisPhD.com, there's a link to the Substack. There is a link to CircuGuard if you want to go buy it. All my social media is there. I've got TikTok videos and YouTube videos explaining a lot of the research that I did. You can email me, and you can join my email list if you want to hear more.
Yep. And we'll, ladies and gentlemen, we'll have that in the show notes down below for you to access easily. Well, thank you so much, Dr. Ellis. You know, your contributions to science in a pragmatic way are something that I think, quite frankly, we need a lot more of to drive forward actionable results from the science we do. I'd love to see more of the grant dollars turn into solutions for people's health than they have in the past.
And that's been a big thing for me, like the past year and a half. It's like, are we doing studies or are we making solutions? Because studies are foundational, and you have to build up a number of studies to create solutions. But at the end of the day, if all you're interested in is studying problems, you're not solving them for people. And that's kind of a problem.
I love that. That's the quote of the day. Well, ladies and gentlemen, thank you for joining us on the Peak Performance Podcast. I'm Coach Kyle, here with Dr. K and Dr. Keith C. Ellis, PhD. Thank you.
Thanks for tuning in to another episode of the Peak Performance Podcast, your #1 resource for today's top health hacks and facts. You can follow us on Instagram at the Peak Performance Pod or on X at PerformPod. You can also check out our website and weekly blogs at peakperformancepodcast.online. Again, thanks for watching, and don't forget to subscribe. We put out a new podcast every Thursday. The content of this podcast is provided for informational purposes only and is not intended for the purposes of diagnosing, treating, curing, or preventing any disease. Before using any products referenced on the podcast, consult with your healthcare provider. Carefully read all labels and heed all directions and cautions that accompany the product. Information found or received through the podcast should not be used in place of a consultation or advice from a healthcare provider. If you suspect you have a medical problem, or should you have any healthcare questions, please promptly call or see your healthcare provider. This podcast, including Coach Kyle, Dr. K, and producers, disclaim responsibility for any possible adverse effects from the use of the information contained herein. Opinions of guests are their own, and this podcast does not endorse or accept responsibility for statements made by guests. This podcast does not make any representations or warranties about guest qualifications or credibility. This podcast may contain endorsements and advertisements for products or services. Individuals on this podcast may have a direct or indirect financial interest in products or services referred to herein.